Hi guys, my name's Cam, and in this video I'd like to talk about how the eye detects light. Light detection. And because this is a little bit more of an advanced foundational concept, I'm going to assume that you guys are familiar with the earlier foundational concepts and know things like how action potentials work and different types of signaling mechanisms in the cell, like G protein signaling, for example, because that's going to be important in this video. So, the organ that detects light is obviously the eye. And like all other sensory organs, the job of the eye is to convert stimuli in the external world into signals that our brains can understand. So for the eye, it's going to convert light signals, photons, into electrical signals. And by electrical signals, we mean the flow of ions, because that's essentially what electricity is. And so this process of converting one signal into a different type of signal is called transduction. And this is essentially the main job of every sensory organ. So let's take a look at how the eye does that. So here we have an eyeball, and on the front of the eyeball is the cornea, and behind the eyeball we have our iris, and in the center where the light enters is the pupil, and behind the pupil and the iris is the lens that allows for light to be focused on a special layer of the eyeball called the let me just change color here, called the retina. And the retina is quite special because it has these special types of cells that can detect light. And these cells are called photoreceptors. And there's two types of photoreceptors, which I'm sure you've heard of before, and they're called rods and cones. And let me make some room here. Rods, just like the name says, are rod-shaped. And cones, there's the nucleus, and cones are cone-shaped. And there's the axon. And in the rods and cones, we have these specialized discs. And these discs are basically lipid bilayers, and here in the rods as well. And these discs are where the light detection occurs. So if we zoom in on one of these discs, we'll see that in the lipid bilayer of these discs, there are a bunch of little proteins. And these proteins are called opsin proteins. O-P-S-I-N, opsin proteins. And in the rods, there's only one type of opsin protein, but in the cones there's different types of opsin proteins because different types of cones can detect different colors of light. And so in rods, it's called rhodopsin. And inside rhodopsin is a special molecule that can absorb photons. And this molecule is called 11-cis retinal. And when 11-cis retinal absorbs a photon, light, photon, it is converted to all trans retinal. And when it converts to all trans retinal, that activates rhodopsin. Now, rhodopsin is a G protein coupled receptor, meaning it's a receptor that activates a G protein. And this G protein, when it gets activated by rhodopsin, this G protein is called transducin. When it gets activated, it binds a GTP. Once activated, it activates another protein in the membrane called cyclic GMP phosphodiesterase. And cyclic GMP phosphodiesterase can convert cyclic GMP to GMP by cleaving the phosphodiester bond in cyclic GMP. Now, why is that important? Why do we want to cleave cyclic GMP? Well, the reason is that in the plasma membrane of photoreceptors, we have these ion channels. Okay? And this ion channel is actually gated by cyclic GMP. So when cyclic GMP binds to this channel, it opens it. And this channel conducts two ions, sodium and calcium. And there's another ion channel, that conducts potassium. And this one is not gated. It's a potassium leak channel. So, when cyclic GMP is cleaved into GMP and we lose cyclic GMP, this channel is going to close. And without the inward flow of sodium and calcium and only the outward flow of potassium remaining, we're going to have hyperpolarization. Now that's a little counterintuitive. When the stimulus comes in, we're getting hyperpolarization, where normally in other types of sensory systems, we get depolarization. But this is important to keep in mind because we do get hyperpolarization in the photoreceptors when the stimulus comes in. So how does this lead to a signal going to the brain? So when the rod gets, although the rod's getting hyperpolarized, it synapses on another cell called the bipolar cell. And it's called bipolar because it has two extensions. And the bipolar cell synapses on another cell called the ganglion cell. And this could have multiple extensions. So, because the photoreceptor, or the rod in this case, is synapsing on the bipolar cell, this synapse is inhibitory. And on the bipolar cell's synapse on the ganglion cell is excitatory. I forgot to name this here, ganglion cell. Okay, so when the rod cell is hyperpolarized, it can no longer inhibit the bipolar cell, and now the bipolar cell becomes activated, and it can activate the ganglion cell. And the ganglion cell can now send an action potential to the brain, specifically the visual cortex. And so this is how the activation, or this is how light can be transduced into an electrical signal. Light stimulating the rod cell activates this signaling cascade from rhodopsin to transducin to cyclic GMP phosphodiesterase, and then cyclic GMP phosphodiesterase cleaves cyclic GMP to make GMP, which then causes the cyclic GMP gated channel to close, and we actually get hyperpolarization. We're not getting depolarization, we're getting hyperpolarization. And hyperpolarization of the rod causes it to no longer be able to inhibit the bipolar cell. Now the ganglion cell becomes activated, and an electrical signal or an action potential can be sent to the brain saying, hey, this photoreceptor has detected a photon. So, I hope that was helpful. I'll see you in the next video.